virtual event, How Biomarkers Are Changing Cancer Care, sponsored by Genentech. Thanks so much for being here. I'm Matt Herper. I'm the Senior Writer of Medicine and Associate Editorial Director of Events here at STAT. And before we get started, a few quick housekeeping items. At the bottom of your screen, you should see several icons. Use the chat feature to chat amongst yourselves or to send a message directly to the STAT team. Please do not put your questions for us in the chat. Don't think, though, that means that we don't want those questions. We want them very much. If you do have a question, please use the Q&A feature to type your question, and we will try to answer it live. In the Q&A window, you should also be able to see other questions that people have asked and upvote the ones that you're most interested in. I'll pay attention to what gets upvoted. We will be taking questions throughout the chat, so please don't wait until the end to submit. You can even start now. This is part of what makes these events fun. Thanks to all of you for being here. Please note that the decisions around editorial speakers, programming, and subsequent conversations are driven by STAT's editorial staff. The sponsor has no input or control over editorial conversation that occurs during the event, and the speakers have received no financial compensation. Before we get started, I'd like to welcome Josina Reddy, MD, PhD, Vice President of Product Development for Lung, Agnostic, Skin, Rare Cancers at Genentech, for a sponsored introduction of today's event. Take it away, Josina.
Thanks, Matt. Thanks for leading this discussion today and for your years of insightful journalism on healthcare issues that matter. And thank you to today's speakers for taking the time to share your insights and for your contributions to cancer care and to innovation. Thanks to all of you who have taken the time to join us today. My name is Dr. Josina Reddy, and I'm the Global Head and Vice President for Clinical Development for Lung, Tumor Agnostic, Skin, and Rare Cancers at Genentech. Today's topic lies at the heart of cancer care, and it's one of the factors that drove me to join the industry. The power of combining scientific knowledge with advanced diagnostics to truly target the genetic drivers of a person's cancer. And 19 years ago, when I joined industry, I was fortunate to work on one of the first biomarker-directed therapies that targeted HER2. And since then, I've spent my career working on personalized cancer medicines that are designed to target genetic drivers. These targeted therapies have fundamentally changed the way that we treat a wide range of cancers. And in this era of personalized cancer care, identifying those biomarkers in a patient's tumor tissue or using blood is an essential step in developing a personalized treatment plan. And for a growing share of patients, that treatment plan might include those targeted therapies that I discussed. For some patients, it might involve immunotherapies that help to engage the body's own immune system to fight their cancer. And for many, it may also involve longstanding mainstays of treatment, such as chemo. But patients can only be matched with the appropriate treatment options if they get a complete personalized cancer diagnosis, and that means getting tested for all relevant biomarkers at the start of their journey. Remarkably, though, a large share of patients don't get the comprehensive biomarker testing that they need. If we look at lung cancer, for example, in a recent study, only about 3/4 of patients with non-small cell lung cancer were tested for biomarkers at all, and fewer than half of them received comprehensive testing for all of the potentially relevant biomarkers using what's known as next generation sequencing. And there are now more than 20 FDA-approved targeted therapies for patients with non-small cell lung cancer. Some of these are suitable only for a small number of patients, let's say 2%, 1%, even less. But if you or your loved one is one of those 2%, these targeted treatment options could really make a difference. And taken together, these seemingly small percentages of patients add up to a substantial percentage of cancer patients who could benefit. We believe that every patient who might benefit from one of these treatments deserves to have that chance. So you might ask, why are many people not being tested when the stakes are so high? In part, it's about the fast pace of the scientific advances and challenges with keeping up. Now, historically, there's been some skepticism about whether the cost and effort of comprehensive testing is worth it, especially when some of the relevant biomarkers are pretty rare. But research now suggests that the cost of comprehensive testing with next-gen sequencing is offset by avoiding wasted time and missed opportunities for therapy. Other barriers that happen with testing can be logistical or cost-related, for instance, ensuring that there's adequate tissue collection or access to blood-based liquid biopsies. Insurance coverage can be uneven, lab capacity and turnaround time can vary, and some of this can be a special challenge in community-based oncology practice where many patients get their care. So in short, it's complicated. It's difficult, but it's necessary to get patients and their families up to speed quickly after a diagnosis so that they can insist on the best possible care. In my personal life, I've recently had several friends and family come to me after being diagnosed with cancer, and I've helped to counsel them on how to ask for comprehensive biomarker testing as soon as possible, and they just didn't realize that they should ask for it. And while the reasons for gaps in testing might be complicated, the case to address these is simple. When testing doesn't happen, potentially life-prolonging treatments remain out of reach for patients. And unfortunately, like many other aspects of our healthcare system, this access barrier falls more heavily in some communities than in others. In an analysis that was done by Flatiron several years ago, patients with lung cancer who were Black, older, or on Medicaid had lower odds of getting next-gen sequencing compared to patients who were white, younger, or commercially insured. And in a study that I mentioned earlier, about 50% of white patients with lung cancer received next-gen sequencing, but only about 40% of Black patients. So personalized cancer care should be a source of better lives for all and not a driver of even deeper inequities. And we need to fix these gaps now. So at Genentech, our goal is for every person with cancer to receive care that is personalized for them. And personalized healthcare has been our mission from the beginning, and today we're advancing it in a number of ways. We're doing this through pursuing an unmatched range of new personalized cancer therapies, but we're also harnessing digital technology and data analytics to personalize every aspect of patient care. And most relevant to this discussion, we're teaming up with our research partners, with patient advocates, with healthcare providers to increase access to comprehensive biomarker testing for all patients. A couple of examples, we're supporting campaigns like Longevity's No One Missed. We're participating in the My Lung Consortium, and we're partnering with patient and provider advocates led by the American Cancer Society's Cancer Action Network to advance policy changes at the state level. This problem is bigger than any one company or component of the healthcare system alone, and all of us in the oncology community need to work together to solve it. When we do, there's little question that we're going to deliver better, more equitable outcomes for cancer patients at a lower cost to society. So in today's discussion, you're going to hear about some of the impressive scientific and technological progress that's being made in biomarker testing. You may hear some scientific debates about the practical challenges that researchers, oncologists, insurers, and others are working on to address this with the latest technology. And we at Genentech are thrilled that this conversation is happening. As always, we'll be listening for any insights that might help us do more to deliver personalized cancer care for patients. There's one thing I want you to remember from this. I hope it will be that faster progress against cancer hinges on equitable access to biomarker testing. Thank you. And now I'll turn it back to Stat and Matt Herper.
All right. Thank you so much, Josina. Now here to join me for the rest of today's discussion, please welcome Anna D. Barker, PhD, Chief Strategy Officer at the Ellison Institute for Transformative Medicine of USC and Distinguished Visiting Fellow, Complex Systems, Arizona State University. George Dimitri, Director of the Sarcoma Center at the Dana-Farber Cancer Center and Director of the Ludwig Center at Harvard. And Suzanne Topalian, Professor of Surgery and Oncology at the Johns Hopkins University School of Medicine and Associate Director of the Bloomberg Kimmel Institute for Cancer Immunotherapy. Thanks so much to all of you for being here. And I want, biomarker can mean a lot of things. We're already seeing in the Q&A, everything from DNA sequencing of tissue samples to your blood pressure. But this really is thinking about cancer in terms of how other parts of biology that we have to measure, biomarkers, distinguish tumors as opposed to just where the tumor is, has really changed the way cancer is thought of and treated and the way that drugs are developed over the past 20 years. So we want to talk about that pretty broadly. And the four of us got together before this and talked a bit about what we were going to talk about. We talked about a bunch of these biomarkers, but I thought it might be best to start with a really clear example of one that works well. And I figured we'd pick a recent one and one that got mentioned a lot when we were talking. So Suzanne, I thought maybe we could explain to everybody here what MSI-high is and why it's important.
Yeah, thanks, Matt. So MSI is microsatellite instability. It's a marker generally of very high mutational burden in a cancer. And my field is cancer immunotherapy. And the reason this hit my radar screen is because very highly mutated cancers seem to be more susceptible to anti-PD-1 therapy. And so this was in 2017 approved as a biomarker for patients with any type of cancer to become eligible to receive anti-PD-1 therapy. So tumors with this genotype are very likely to respond to anti-PD-1. So this was the first FDA approved biomarker that crosses all cancer types, and it gets to your point earlier about cancer biology playing a very large role and going beyond simply looking at the anatomic location or the site of origin of cancers.
And so contrast that a little bit with the other biomarkers you have in immunotherapy. Let's talk about biomarkers in immunotherapy. So the PD-1 inhibitors, for anyone who doesn't know, are the big common kind of immunotherapy drugs, Keytruda and Opdivo being the big two, I think.
Mm-hmm. And so in terms of contrasting the MSI-high marker to a marker like PD-L1.
I was thinking exactly like a marker like PD-L1, which was kind of the original immunotherapy marker.
Mm-hmm. Yeah, so they're very different biomarkers, but both are FDA approved to identify patients who are candidates for anti-PD-1 immunotherapy. So a PD-L1, in contrast to MSI-high, it is a marker that can change over time, and it's a marker that can have different levels of expression in different sites of metastatic disease in an individual patient. So there's a lot more biological variability, which has made it more difficult to find the right place for this biomarker, to find the right way to use it effectively to inform cancer treatment and care. So MSI-high, it's not going to change over the evolution of a cancer, and it's not going to change in different metastatic sites in an individual patient. It's going to be there. And so if you could test for this 10 years before you think about giving immunotherapy to a patient, you can go back to an old tumor and find this marker, and it can then help you with your strategy for planning out treatment for that patient. But PD-L1 immunohistochemistry is very different than that. So PD-L1 has not become an across-the-board biomarker the way that MSI-high is, but it has been shown to be helpful in certain cancer types and certain disease settings for identifying patients who are more likely to respond to anti-PD-1.
Now, George, you mentioned this MSI-high as a biomarker that particularly impressed you, and I know we've talked about different biomarkers going back, fitting with targeted therapies going back many years.
Yeah.
What stands out about this one?
Yeah, one of the things that Suzanne's alluding to is that the MSI-high, as she said, it's hardwired into the cancer. It is not going to change. It's locked in. Whereas a lot of other so-called biomarkers, they come and go. The cancer wants to put it on its surface and show it to the internal environment, or it doesn't. And that's the problem with something like PD-L1. Also, mechanistically, MSI-high.
Even with the biomarkers, the cancer's outsmarting us.
Yeah, absolutely. It's using every tool of evolution to change its shape, to change the way it shows itself. It's a pretty sneaky little critter there. The important thing about MSI-high, it's also mechanistic. It was what's so cool. We talked 21 years ago, Matt, about imatinib or Gleevec. And if you got a mutation that turns on an enzyme, so it's not stopping, if you can stuff something into the mouth of that enzyme and it can't do anything, it might stop the cancer. And that's kind of what Gleevec does. With MSI-high, what it is, it's a low fidelity replication system. So the cells are making copies of each other, but each copy starts looking weirder and weirder. And the immune system recognizes things that don't look human. It's why our immune system is good at fighting off bacteria and fungi, and when provoked with the right mRNA, COVID. So I think that's the issue. It can be very mechanistic. And when you see a mechanism-directed biomarker, it's usually much more reliable, much more powerful, and means a lot more for patients because you can really count on it, and it can help us pick the right, very reliable, active drug or therapy for that patient.
So Anna, I wanted to start with this to give everyone a clear example of what an ideal biomarker is. But looking back at, so George just alluded to thinking about this starting with talking about 21 years ago, when that would have been Gleevec and GIST, which is a gastrointestinal tumor. How would, if you could have a conversation with yourself 21 years ago, what would you 21 years ago be surprised about in terms of where we are at finding these kinds of biomarkers, at finding things like MSI-high as we develop new medicines?
That's an interesting question. It does. See, actually, 2002 sounds like yesterday almost, the way things have been moving in this whole space. But so I was pretty familiar with the BCR-ABL gene, and I was very familiar with Gleevec. And I think the assumption then, and actually it sort of paved the way for what we talk about today in terms of precision oncology, is that everyone thought there was just going to be this rampant, sort of numbers of fusion genes, that the BCR-ABL gene was going to be replicated thousands of times, and we were going to find lots of fusion genes, and that didn't happen. But it's happening now. In all of those years, we've been looking for fusion genes again and again and again, and now we're beginning to see how important they really are, because now we're talking about fusion genes all the time, once again. But I think,
Let's explain what
11 years ago, I would have started to say, whoa, we're going to find a lot of fusion genes pretty quickly. Not so much.
Let's explain what a fusion gene is. That's the gene that's kind of involved in the cancer gets fused to something else. Literally, it's literally genes get fused together, and the cancer is using that to turn something that the body normally turns off on all the time. And so there are other examples in biomarkers, right? I mean, there's ROS1 is a fusion.
ROS1's a fusion gene, yeah, and more we're starting to test for them, and we're starting to find them. And it turns out that you can find them with next genome sequencing, but it turns out it looks like it's going to be better, with transcriptomes. So I think we're entering. It's almost like Back to the Future, as you've just kind of put it on the screen, and that is, where we were 21 years ago is where we would like to have been, 21 years ago is where we are now. And I think we're just starting to really see the power of these, combinations. I mean, MSI-high is a very complex biomarker, extraordinarily. And in the sense that it's, as George has just said, it's conserved across cancers, because you're talking about repairing DNA. And that's absolutely, as he said, hardwired into every cell. And so, now we're beginning to see these complexity of biomarkers, I think, is the future. I mean, I think we're, just as you have MSI-high and you have biomarkers like PD-L1, we see the same thing in breast cancer or colon cancer, whatever. We're seeing these complex biomarkers come together to tell a bigger story.
And this is, Matt, let me just clarify one thing too. And one of the things you said is that we didn't see the fusions to a great extent, but transcriptomics is showing us that. Let me just explain that for people who aren't, PhDs out there. When you only look at DNA, you can find DNA from dinosaurs. It sticks around forever. It's easy to understand. But DNA turns into RNA, and then the RNA turns into proteins. The RNA is more evanescent. It comes and goes. It's more fragile. But that's really where it's easier to find these fusions. So the adage of a carpenter is only as good as her tools really applies here, because we didn't have great technologies to find these fusions when all we were looking at was DNA and old technologies. But the new way of looking at RNA makes it easier. And actually now we can even find it in other ways where the technology's gotten better. So I want to point out that I agree with what you said, and we're in a very different space technologically, even in the past 20 years. Like my iPhone's better than it was in 2007, and the technologies we're using for biomarkers are a lot better than they were in 2007.
We were all blown away by the technologies we had, I think, to a person here, right? Back in 20 years ago. I mean, we could sequence a genome. There was there was Gleevec. We were starting to learn about the EGFR inhibitors, which are another important. So why are we sure that we have the right tools this time? How do we know that we've actually invented the wheel?
Maybe I could speak to that. So in 2004, after we had published the first, sort of human genome sequence, Francis Collins and I sat down one day and said, "Isn't it time to sequence the cancer genome?" Plural. And we started with Sanger sequencing, you'll remember, all the way back in 2004, with an ideal that we would hopefully be able to sequence 20 cancers, 10 cancers, I think was where we started. We ended up sequencing 30 cancers, but during that period, actually, Matt, we saw us go from doing very simple Sanger sequencing, well, sort of simple, but not that simple, all the way to next genome sequencing. And during that period, we also brought in, sequencing the transcriptome, as George just mentioned, and even the methylome. Getting all the pieces of that very complex sequence together was really, I think, quite an accomplishment. And since then, now we've got, nano constructs to do this, et cetera, et cetera. So technology is moving very quickly now.
So a great question from the audience. I want to start. We have 15 audience questions already, so I want to start threading them in. But from Raed Samara, which is, where does TMB, which is tumor mutational burden, sit nowadays as a biomarker for immunotherapies, and how does it compare to MSI status? And Suzanne, I was wondering if we could talk about this a bit as a contrast to MSI-high, but also to the difficulties in searching for these when we have new drugs. Because there was a lot of excitement about TMB, and I feel like that's waned a bit. So walk us through the TMB story.
Yeah, so I think, as we gained more experience and information from testing immune checkpoint blockers like anti-PD-1 in different cancer types, and pulling all the information together, people started to realize that it was the cancers that tended to have more mutations in them that were more likely to respond. And, a great example is lung cancer in smokers. So those cancers are caused by a carcinogen, and they tend to have high mutational burdens. And they are also more responsive to anti-PD-1 than a cancer like pancreatic cancer, which has a very modest or low tumor mutational burden. So putting a vast amount of information together about, 7 or 8 years ago, maybe longer, people started to realize that there was this association of a high mutational burden in the cancer with a greater likelihood of response to anti-PD-1. But then, in terms of developing that knowledge as a biomarker, it required establishing a threshold, like a cut point above which you would say, if this tumor has so many mutations, it's going to respond to anti-PD-1, and that would qualify the patient to receive anti-PD-1 therapy. So that's where the problem came in, is that it was very difficult to define where that threshold is. Eventually, the FDA did approve one anti-PD-1 drug, pembrolizumab, for any type of cancer that had a TMB that was higher than 10 mutations per megabase in the tumor. So that's about 300 mutations per tumor genome. But there's now information that maybe one cut point like that is not going to be the best for every cancer type. And so this has, now started an ongoing conversation in the literature about whether a certain type of cancer, needs a higher cut point or a lower cut point in order to use this marker, in the best way. It's still an unresolved issue, but nevertheless, we do have that FDA approval based on the cut point of 10.
And I just wonder, thinking about this, of what we've mentioned, most of the MSI-high works for all cancers, but MSI-high is rare. right? That doesn't happen very often. And we also we started off talking about Gleevec, which is the classic example of that. Is a BCR-ABL driven cancers are rare, and GIST, which you were treating, it was only with specific biomarker, and that's also rare. Whereas immunotherapies are really the current set of drugs that are being used. You know, you look at the labels for these drugs, and they seem to list every single cancer you can think of. Is this kind of search something that's going to, how helpful is it going to be for common cancers? Are we going to be able to find biomarkers that determine how you treat people who have those common cancers?
I think yes. I mean, let me just go out on a limb. I think yes, because I think there are some big bumps on the road to get there, though, Matt. But if you look at something like HER2, let's go all the way back to HER2. You know, there's something that we're using.
Target for Herceptin, which was really the first of these we did.
Absolutely, it was the first.
Positive HER2, you get this drug and it can treat breast cancer in a way you couldn't before.
Right, and just think back, going back to to something similar to the BCR-ABL story. This is one where it turns out that one way or another, it's helping almost every single patient. And, I think there are going to be biomarkers like that. This is a receptor, obviously, and if it's amplified, you're going to be in one class of patients. We're starting to use these biomarkers very effectively to subtype these diseases. So when I say I think it's going to work well in the future, I think you're going to use it in different ways. And I think you're going to start using the biomarkers to subtype. And when you start subtyping tumors, you do much more effective treatment. So I think maybe not single biomarkers, but maybe biomarkers that actually you can use to subtype patients, like we're doing in breast cancer now, like we're doing in a lot of cancers today.
And breast cancer really set the standard for really in one of the first biomarkers. Did it have a hormone receptor in it? And should you use hormone blocking therapies to treat it, right? That was a biomarker that saved the lives for millions of women around the world with hormone therapies. So in other words, our field didn't just get started in 1998. This field of trying to target the root cause of what's driving a cancer cell differently from a normal cell is something that's part of the discovery mechanism. And as we get better, I'm with Anna, I think we're going to find more and more reliable biomarkers. They're probably going to have to be multiple. They will point to combinations, almost certainly, because most of the common cancers are pretty complex things. They're not simple like GIST with 1 or 2 driver mutations. They're not simple like HER2-driven breast cancer, which has that dominant HER2 focus driving the cancer. They've got all sorts of little things driving them, some of which don't stick around. They can change, just like you said, Matt, just like you said, Suzanne, that they can shift. They can use this epigenetic shifting to turn from blue to green. They're chameleons. And I'm not worried about that because I do think our technology will catch up to it and we'll have 3 or 4 combinations, just like we do for many severe infections. Look at multi-drug resistant tuberculosis. You still can come up with multiple drugs to overcome that. And that's my model. My model is still infectious diseases. That's where we've got to go in our field.
Suzanne, how do you think about this from an immunotherapy perspective, though? Because I do feel like with the immunotherapy drugs and at least some cancer indications in non-small cell lung cancer, the learning has been that maybe you didn't need to be as selective as people thought you would be with them. That especially if you can find something like that may be working synergistically with the immunotherapy. So,
yeah, I think people have that attitude now because the biomarkers that we have available are not as strong as we want them to be. So what you're referring to is that there are some patients whose tumors are marker negative, and yet they can still respond to the drug. But for that reason, I think some patients are probably being overtreated or treated with a drug that's not as effective as it should be for them. So that's really our goal now is to define markers or combinations of markers that are going to be a lot more sensitive and precise than the ones that we already have.
So a question that I wanted to get to from Rajarshi Sengupta. Can you comment on the clinical utility of using one biomarker versus a panel for making treatment decisions? And he lists MSI, TMB, HLA, PD-L1. Are we moving to a world where this has been an area of tension, right? I mean, I think even in non-small cell lung cancer, which I think of as a very biomarker rich, there are some people who are sequencing and looking for kind of anything, and there are some people who are doing immunohistochemistry for ROS and ALK and the kind of big EGFR, the big ones we know. Are we moving to a world where everything is a panel in every cancer? Is it going to vary by cancer?
I think that's the direction that things are going, is that people are, right now we have unidimensional markers for immunotherapy, and we're trying to develop something that's deeper than that. And it turns out that some of the markers we have do not depend on each other functionally. So for instance, PD-L1 expression does not depend on TMB. So that means that you may have more power by combining those two markers in a single test. So there's a lot of work going on in that area now, but so far, no home runs, nothing's that has made it to an approval.
But I think, Matt, though, if it's interesting, if you kind of look back to one of your original questions was, let's look back over time, and there's a huge shift in oncology today. We're collecting enormous amounts of data from all kinds of, especially from molecular profiles on patients, and you wonder if eventually, and in parallel, we're collecting real world evidence, real world data, and as those come together, the future may be that, maybe the genes are going to tell us from what we see in the data. And, we're going to be able to go back and start to construct these panels prospectively per cancer. I really wonder if that's not the future, because if you look what Suzanne is doing in immunotherapy is an entirely different kind of biomarker. I mean, the kind of biomarkers that we're measuring for immunotherapy are nothing like the precision that we're trying to get in oncology with single biomarkers, let's say. She's dealing with a system. I mean, she's essentially readjusting a system when she's actually giving immunotherapeutics. And that's the future, I think. I mean, I think it's going to be figuring out how to deal with these in a much more systems orientation, where we're putting these things together based on the data and the advanced analytics we can use to actually determine maybe even prospectively what we should be using.
Let me comment on that too, if you don't mind, 1 second.
Well,
Matt,
I actually want to go deeper on this. The audience is asking about it, so go.
Yeah, because when I trained in 1987, lung cancer was just lung cancer. There are two kinds, little cells, big cells, but the big lung cancer cells, so-called non-small cell, was just all treated the same way. We didn't understand anything about it. Now we know it's at least two big types of diseases, right? Those diseases driven by specific mutations, EGF receptor, ROS, ALK, RET, you name it. There's a few KRAS. They're kind of things in there driven by mutations. On this side, though, are still those things we don't really know. They're just bad, but that's where the immune system can really hit it, because they don't look almost human, those cells. And if you jazz the immune system up, you can really get those patients some benefit with chemotherapy as well. Whereas on this side, if you've got an EGF receptor mutation, chemotherapy doesn't add much and immunotherapy doesn't add much. So as a practicing physician, the key thing is to say, which bin is my patient in, this bin or this bin? And that's really important, because the sequencing can tell you something about both. You can get an idea of the tumor mutational burden from looking at the DNA, and that'll give you a hint as to whether that TMB is high, and you might head towards immunotherapy if there's no other so-called driver mutations. But if it's got an EGF receptor mutation, if it's got RET or ALK or any of those other things that are highly effective when targeted, you definitely want to be on that side of the bin and not use the immunotherapy as your first line of attack. I think it's kind of easy.
It's kind of, in one case, you've got a switch that you've got to flip very carefully. And the other one, you've got a tumor that's a monster that you just have to beat up, right? I mean, because there's any one switch isn't going to do much.
And somebody in the chat mentioned how important it is to get turnaround time. You know, if you want to put your patient into a bin and come up with the right shot the first time out of the gate, you do sort of have to think about that test, order it, and get those results back pretty quickly. And that's why a lot of academic labs, a lot of commercial labs focus on turnaround time when it's an important thing to help drive a decision in a patient about whether you use immunotherapy, targeted therapy, chemotherapy, or some combination. So that's important.
Let's pick up on that, because I was going to get to it later, but Su Chang in the Q&A asked about how do we get this to underserved populations? How do we get this? You know, I know from people that I know who've had cancer that if they go to MGH, George, or if they go to the Farber, they get biomarker testing. I'm sure that's true at any academic institution, but there's a lot of people in the community who aren't. What can we do to make sure that this is standard for people who could benefit?
I mean, this is where data has to rule. We should practice evidence-based medicine. And if you need that data, if you need that evidence to make a decision for your patient, just picture what you would do for your family member, for your loved one. Everybody can picture that. If I cannot make a wise decision without having those data, how can I make a decision? And then we as professionals have to justify that mainly to payers. And the payers will say, well, it's a lot of money. Why should we pay you? And then we have to have a strong enough evidence base that it really is a life or death, significant prolongation of life versus closer death that really drives that to a high quality healthcare system. And we're judged on that. That's my bias.
But, you know, but George, when things work, I mean, like Gleevec, for example, I mean, patients get Gleevec today and they also get tested. So I think, Matt, when something obviously works, then, you know, then CMS steps up to pay for it, and people in the community hospitals are going to use it just like people in the academic medical centers. I think our problem with biomarkers is there's been a long sort of regulatory kind of learning curve here, and learning curve on both sides of this equation. And I mean, now we've even got breakthrough status for certain diagnostics now. I mean, so this whole field has moved very quickly. But your point, I think, is the point almost to take away today. This has to be very broadly distributed to the 80% of our patients that are treated in community hospitals and in all kinds of other places other than our cancer centers. And I'll say one more thing about this. You know, the White House is talking today about the sort of reigniting the moonshot. And Eric Leonard was speaking yesterday to some of us about we need a learning system in oncology. We need a learning system for biomarkers. I mean, you know, you take any one of the diseases we've talked about today, we could actually put that data set together for, you know, for PD-L1. You could have a national biobank for some of these kinds of things, or a data repository for these tumors. We just haven't done it. Creating these learning systems could change a lot of what we're talking about. And it would allow community docs to actually just orthogonally look up their patient in the right data set. That's, you know, those are kinds of things. Those are not intractable problems. We should solve these problems.
Well, they're not intractable, but it sure has every time they seem tractable, every time they seem tractable, they don't get
traction.
They don't get traction. That is exactly what the construction I'm looking for. How do you guys feel about, I mean, how close are we to being able to collect those data in real time and really know find biomarkers emerging from kind of this soup of genetic and transcriptomic and protein data and actual patient records? I mean, how close are we to being able to do things like that, or even being able to run the trials faster that we do now?
I think we're close. Honestly, I do think
go on. You're close.
I think we're very close.
I think we're really close. I think the academic labs and the commercial labs are currently sort of trying to better one another, because the academics want to stay alive, the commercial labs want to take over. And it really is almost an arms race of who can do it better. That's a positive cycle, I think, of competition to try to see how fast from the minute a sample can get out of a hospital into a pathology lab, a molecular pathology lab, how fast can a high quality annotated result be given back to the doctor? And I think that's it. We have a problem also, let me just point out, it's not just that technical element. We have a huge education problem, because a lot of doctors were trained before a lot of these data were available. We have to retrain everybody to know what they really mean so we don't see a massive epidemic of misinterpretation. And I think that's another element. It's where we bring in our patients. And I'm so glad to see so many patient advocate groups here in the chat, because patients are really their best advocates, and they can be the ones to share their understanding of a biomarker or of a disease or even of a term of art with other patients so that they can use it wisely, because you can't count on all the doctors being up to speed as much as this field is changing. That's a big fear of mine. I think that's where the academic centers need to focus on that education as much as we can as well. And Suzanne, you saw that with the advent of immunotherapy, how fast you and your colleagues in melanoma adopted it in a really smart way. It was wonderful.
Amazing.
So, so far we've been talking about biomarkers to launch a treatment in a patient, but maybe should we also talk about biomarkers to help us strategize while patients are on therapy and select next treatments in case the first one is not working. Maybe blood-based biomarkers are going to become important in this area, circulating tumor DNA and so forth, and giving us earlier information about when relapse is occurring and allow us to, get a jump start on what the next treatment will be.
How much are you that now, Suzanne? I mean, that liquid biopsy is the term that people seem to get for this, but being able to monitor cancer through blood is a big deal. How much do you feel you're able to do it now, and how much do you expect that to change over the next few years?
I don't think this is standard practice, certainly for most cancers, but there is a lot of study in this area now. And, one scenario is a patient who starts out with a positive marker in the blood, and then is on a treatment, and you want to see that marker dropping so you can follow the trajectory of the marker over time and get a sense for what the outcome is going to be, or looking to see the marker completely disappear and see if that correlates with prolonged survival. I mean, these are all the kinds of studies that are in process now that, hopefully soon we'll have some of these answers. Well, one of the challenges with blood-based markers like ctDNA is the sensitivity. I think, we're in the laboratory still striving to improve the sensitivity so that we could pick up, for instance, a tumor relapse after surgery when the tumor is still at a, microscopic or very small volume.
But Matt, I think if you think about biomarkers, we're all focused on treatment and and selecting these patients for treatment. But this is probably the highest value proposition in all of oncology, all the way from diagnosis, to prognosis and predictive biomarkers, which is kind of what we've been talking about. But there's also treatment planning, and then there's monitoring. I mean, at every phase of the patient's journey. And it's the patient's journey we care about. It's all the way from diagnosis to treatment, and then even aftercare. And more, we're thinking even about that period of a patient's life. And especially, our survivors think a lot about that. So, I mean, I think these biomarkers we're talking about have application across that entire space. And more, there's going to be all kinds of approvals for these biomarkers for different applications. And the FDA says your biomarker has to be fit for purpose. That means it has to have context. In other words, you have to know what you're going to use your biomarker for. And right now we're focused on, on treatment selection, but there's going to be a lot of biomarkers coming to manage the patient's entire journey from diagnosis and pre-diagnosis, actually, all the way through survivorship.
Yeah, and I'd like to stay away from broad, expensive screening of the blood in people who are otherwise healthy, but I do want to focus on one little area that I feel very strongly about, which is so-called cancer predisposition syndromes, where cancer really runs in a family. The first famous one was Lee-Fraumeni syndrome or Lynch syndrome, driven by different mechanisms, but we know that those are bad. Lynch syndrome, for example, is again a low fidelity copy problem. The cancer cells start and they keep making weird versions of themselves. And it's really important because a lot of those young Lynch syndrome patients we think can be cured by immunotherapy. They can certainly live a long time. I know at Hopkins, Liz Jaffe has an advocate down there who's very vocal and very an amazing, inspiring story of her fight against Lynch syndrome and colon cancer that is done so well with immunotherapy. And there are many other such patients. I just want to point that out because one biomarker in cancer predisposition families is looking at the genes that run through the family and seeing if there is a predisposition and then monitoring those people really so that we can get a cancer as early as possible where we know they have 100 to 1,000-fold higher likelihood of developing cancer than the general population. I do want to stay away a little bit from the, let's just give everybody a genetic test.
But stay away. Do you mean you don't want to talk about it now, or you're not really comfortable with the idea of using a liquid biopsy screening for the general populace?
Oh, I'm very comfortable with it. I'm just not comfortable with today's technology that it will be specific enough. I worry about all the false positives. So I love the idea that it's research.
Where half the people don't have cancer who are told they do.
For example, yeah.
We're going to be, I think, Matt, you're going to mention this later. We're going to be talking about that. We have a forum at my institute, and that's a topic we're taking on next week. Actually, I think it's probably one of the most interesting and exciting events in all of oncology, and right now probably the most problematic in terms of the pros and cons. It's a very interesting area.
I mean, for other use. Asaf Vestin's asking this. I see it a few times in the chat. What are the challenges to doing all of this more routinely through blood tests as opposed to having to get tumor samples? Is liquid biopsy standard of care now? Does it get there?
I think Suzanne said it. It's about sensitivity, not so much specificity. If you see a mutation in EGF receptor,
George should mention tell them the difference between sensitivity and specificity.
Yeah. Sensitivity is, can you pick it up? If you don't see it, is it not there? And specificity is, if you see it, is it really there? So I think liquid biopsy is, you can count on it. If you say you've got a receptor mutation, you've got an EGF receptor mutation, I believe it. Most of the good labs, academic and commercial, they're not going to make a mistake. They could still, but generally, if they report something, it's there. The hidden secret in our field is that if they don't see anything, it doesn't mean it's not there. In other words, a negative is not negative. A negative is non-informative. You haven't learned anything. So if that test cost you $3,000 out of pocket, you just wasted your money because the technology is not great. Now, in two common cancers, lung cancer and colorectal cancers, those tumors do tend to shed a lot of DNA into the bloodstream. Nobody really understands why, but it's so much easier in lung cancer and colon cancer to really detect a lot of circulating tumor DNA in a useful way. I actually think technology will get us here. It's a little bit about detecting that molecule of DNA and analyzing it, but it's also a little bit about the mathematics of figuring out how to analyze these big numbers and what's the likelihood that you made a mistake in a calculation. So I'm not going to get into that, but I think the world has learned a lot about the Theranos debacle because things that sound too good to be true probably are. And I think we're still at the point where liquid biopsy is really good, but it's not as good as maybe people would like it to be.
I wanted to just mention for people who are thinking about this, this is the same problem we're having with COVID tests right now. Yes, are they sensitive or are they specific, right? And if you want to know if you have COVID, you want a really specific test, which is what the FDA has pushed the rapid tests on. If you want to test everybody who's showing up for Thanksgiving dinner, you might actually want sensitivity so you can be sure that you get anybody who's carrying COVID. So this is the problem with every diagnostic test. I have two really great questions, and we've got about a little less than 10 minutes. So I want to all of you both of these are from the audience, and just go through Suzanne, Anna, George. The first is, what are some of the new biomarkers you're looking forward to over the next 5 to 10 years that you're most excited about, and why? And if you want to pick a technology instead of a specific biomarker, but just basically, what are you excited about going forward that we haven't talked about? Suzanne?
Well, I'm excited about the potential for multidimensional or multiplex biomarkers that we were talking about earlier. And there are some technologies that wrap this all into one package. So one would be gene expression profiling. Can we find a group of genes whose expression, either up or down, can be packaged and will tell us that a treatment might be effective in that particular situation. Another technology is done on tissue slices, which is a multiplex staining of different proteins in the tumor. It's called multiplex immunofluorescence, which is something that is being pioneered here at Hopkins and in other centers. So these tests can give us a lot of information all in one package, and maybe more sensitive and more specific for the purposes that we need them.
And I think, Matt, I'd like to see much better biomarkers, highly effective biomarkers for the big killers. I mean, those cancers that we haven't been able to do much about, pancreas, my own special interest, glioblastoma, these are cancers that, what are the unknown unknowns here? What are we missing with these cancers? We can't seem to detect them early enough to actually stop them. I'll say something about technology, though. I like to look over the horizon, and I think that we're going to have the capability even with indwelling sensors one of these days to be able to detect cancer well before it's ever going to be clinically diagnosable. And, if you had the ability to do that and you could deliver drugs and monitor patients in nanoconstructs, let's say, I think that's all doable. I think it's over the horizon, but I think it's all doable. And I think biomarkers will be key to actually doing all of that. So I'm very optimistic about, as you can tell, about biomarkers.
I know Anne has a particular interest in glioblastoma, the most lethal form of brain cancer. And it is a very informative example because it is a disease we've done very little with, except now we're starting to see some exceptional responding patients to immunotherapy, but only a few. So we need to learn why those few people, why are they so lucky, what's different about their tumor. And it offers me, I'm not a brain tumor specialist, but there's a guy at our place named Brad Bernstein who did a brilliant piece of work by looking at so-called single cell sequencing. He looked at a bunch of brain tumors and took each individual cell out and looked at the DNA in each of them. And brain tumors traditionally were characterized as different groups. You've got this biomarker type of brain tumor, you've got that biomarker type. And by looking at the individual cells, Dr. Bernstein realized all brain tumors have everything. They're just all there. It's a much worse enemy than anybody could have imagined. So, that also though gives us a chance to say, okay, good, then how do we jazz up the immune system to beat that beast? If it's that bad, we must be able eventually to find some orthogonal technologies that might go after it. So again, some of it's biomarker, but some of these biomarkers can help push a specific therapeutic research strategy ahead. And I think that's what that did to some extent.
So the other question that I got from the audience that I really love is from Brack Self. And if I wanted to start from zero and learn as much as possible about biomarkers and cancer treatment at an advocate level, where would I start? I was hoping there might be some recommendations. One is obviously tune into Anna's session on the 10th, which the link for that will be in the chat. But what else would you recommend to patient advocates who are seeing this alphabet soup of biomarkers, all these technologies we've talked about, and are trying to figure it out?
Maybe I could start. At AACR, we have a lot of great advocacy programs, most especially our Science of Survivor program. And it turns out that advocates are the new scientists when it comes to biomarkers. They actually will go to their, once an advocate understands that this is important in their disease, they're not going to leave their physician's office until they get a test for that biomarker. And there are even groups, like there's an ROS group, that's actually formed around that particular biomarker. So, I think it's yes, ours, it's an ROS group, for lung cancer. So, but no, I think you can connect to lots of these groups like AACR and ASCO who have these training programs, and they will, they're really going to keep you up to date. and also, read the literature. I think that patients are the smartest of all the people I know in the biomarker space. They tend to know the most about their biomarker.
Let me add to Anna, for the unusual fusion that happens a lot in sarcoma patients called NTRK, there are these NTRK fusions,
Yes.
which also happen a lot in lung cancers and happen in colon cancers and cholangiocarcinomas. They happen everywhere, brain tumors too. There's a group called NTRKers, and they're really trying to teach each other and actually impact public policy around how should people be made aware, how can we make it aware to insurers that this is life-saving and that it's really something that's non-trivial. And then they're pushing technology because they know that if we come up with a faster, cheaper way to find the fusions, people will have better access to it. So again, technology is the future no matter what the question is, in my opinion. Then use of that technology has to be wise. I also think there are some general white papers that AACR and Anna, you, even when you were at NCI some years ago, you were behind many of those white papers on biomarker qualification. What is a good biomarker? I would recommend them to my students and to anybody listening. So just look through some of AACR archives, and maybe Anna, you could suggest some of those at your meetings or to Matt. Suzanne,
With what's been said, really all the major oncology meetings now have big sessions about biomarkers, and maybe some of this is going to be too technical for a general audience, but organizations like the AACR or ASCO do have patient advocacy tracks that I think would be very useful to attend.
All right.
and real advocates can get to those big meetings for free very often. The professional societies are recognizing the importance of partnering with advocates, which I think is long overdue, but they're doing it for the past few years. So I think that's important.
And I guess the big thing to emphasize there is don't be afraid of the literature.
Right.
Okay, well, we are at time, even though I have 3 more questions from the chat I want to go to, but I think it's time to wrap up. So thanks to all 3 of you, to Anne, George, and Suzanne, pioneers in your field. Thank you so much for doing this. It was great having you. And the Ellison Institute will be hosting an Insights Forum on early cancer detection and prevention, as we mentioned, on February 10th at 10 a.m. Pacific time, 1 p.m. Eastern time. So check out the chat for the link to register to that. You can join us for our next event, the Exhaustion Epidemic, examining the COVID-19 burnout crisis in healthcare, on Monday, February 7th. My colleague, Nicholas St. Fleur, our Associate Editorial Director of Events, will be sitting down with experts to discuss the emotional burden healthcare workers are facing and uncover the solutions needed to support and retain them. I know all the doctors I know are burnt out, particularly the ones who just see patients. So this is a really important topic. More information can be found on the STAT website. Thanks again to everyone for being here. Thanks to all of you for coming. It was great to see such great turnout. Stay safe and be well.